Thank you for joining us today on our speaker series. I am pleased to introduce and hand the reins over to our guest today, Richard Parent, who works with a company called MustangBio, and he's going to share a bit about his background and about the work he does. And then towards the end, we'll engage in a Q&A session. I encourage you all to use the chat feature. If you want to enter your questions there, I'd be happy to ask them at the end. And feel free to raise your hand or ask any questions as we go. So over to you, Rich. Thanks for joining us.
Well, thank you. Thank you, Helen, for the introduction. Just want to throw the screen up here. How's that? You guys see that okay?
That works.
Okay. Let's see if I can get the pointer now. Okay, you can see my pointer, right?
Yes.
Okay. All right, so thank you. And I was asked to sort of give a talk. It's pretty open-ended, and I figured I would give a little glimpse into my efforts and hopefully possibly some of yours. My goal in this is to deliver an engaging presentation, which I hope is the beginning of something new. I think every presentation in science is something, at least ones that I've delivered, is ones that sometimes they're new beginnings for people who listen to the presentation, and or they're new beginnings for me because of the questions that are asked. So I never look at presentations as sort of a culmination of my efforts. I always look at presentations as being the beginning of something new on the other side. And so I have a challenge. The challenge is to formulate a question. I have found that question formulation is extremely important, and it helps with engagement. And I'll tell you that when I was freshly out of college, I used to go to this meeting. The Harvard had these immunology meetings every Wednesday, and I would trek over there from Boston, where I was working at the time, just to be able to go to these meetings and listen to these world-renowned speakers in an area which I was very, very interested. I never thought of myself as being one who was a really good question maker, but it just so happened to coincide with a paper that I had read, which asked a question about, what's the difference between people with really high IQs and people who are just of average IQ. And one of the things that I recall was that questioning was a very big difference, and that people with high IQs questioned more frequently than people who had average IQs. And so that was interesting to me because I was going to these Harvard meetings fresh out of college, and I was finding that I was not retaining a lot of the information because they were speaking on a different level than I was quite honestly used to. And it was a forum like you see here, very large presentation, and it was one where it's very intimidating as well, and that I would never ask questions like I would have, in a normal classroom setting. But I found myself making a goal where it was to ask a question. And in doing so, I realized that I began to actually understand more of the information. Because in order to formulate a really good question to ask, you really had to sort of pull together information. It's not like entertainment when you go to the movie theater and you just sort of let the experience pass through you. This was one where you were constantly asking questions and trying to pull together different points that were being made during the presentation. That was very helpful, all in a way to be able to develop a question that I wanted to ask. And in the end, I ended up learning more, I think, during that process. So I've always had a really big thing about asking questions. So I want to step back a little bit. I want to tell you that I grew up out in Chicopee, Massachusetts, And once upon a time, I sat in a seat just like this, probably the same one you're sitting in now. This is the old Chickadee High School. If you're not familiar with Chickadee High School, it looks a little bit different. In college, my experience was I was the first in my family to go to college. So I started off by going to Holyoke Community College, and I decided to focus on a degree in biology. Back then, I thought I'd become a teacher because I love science. I love life science specifically. And the only career I really saw around me, other than Quincy, anybody remember that, was being a high school biology teacher. And so I always thought that that's what I was going to be heading towards. That was until after I graduated with my associate's degree, I went to the University of Massachusetts. And there I found a really small department, so it felt like a small school. And I liked it so much that I decided to stay and get my master's degree. My experience, however, in research really began to flower when my senior year. One of the things that you can do is you can ask to do a senior research project. And so I did that with Dr. Wong, and she was great, and she really sort of captivated the whole idea of perhaps there's a career here in doing research, because I hadn't even heard of biotechnology at the time, and it was sort of a blooming area, a career choice that I hadn't even considered before. So I decided to stay and went for my master's degree, and in doing so, I also had to develop a thesis. And so this is really sort of my first time formally at actually developing a project. And so something that I've always gravitated towards in talking about projects is first using the impossible triangle to describe a possible project. Of course, at the very and the point I'm trying to make here is that there's basically three important parts to this. There's you, there's your teacher, your advisor, parent, or your boss, all of whom, if they're really good, their main intention is to see you succeed. And so, and growing up, it's usually just those two people involved in lots of the things that I would do. That is until I had to do this thesis, in which case I had an academic advisor who helped guide me as far as finding people who I could contact and to start developing a project. And so he came up with an idea and pointed out to me that over in the chemistry department, that there was a professor working on an area which might be interesting to me. And so that started this triangle for me. I bring this up because this is also really important, I think, and some of you might be interested in developing projects for the science fair system. And while lots of times it's your teacher and you, it can also become your parents, some other professional. And so there was a time when I was a teacher, and I made students do some research. They would write letters, and you would be amazed at how many people, all around the world would be interested in assisting high school students. There are grants for which they're working on oftentimes who have money just for outreach programs. And oftentimes they have materials that allow you as a high school student to actually, get a running start as opposed to trying to generate things that just the normal high school student wouldn't have access to. And so, this is my estimation. I think it's one of the best ways to develop a project. And so I wanted to go into my research project real quick and give you some background into the first project that I got involved with. And so I remember my academic advisor pointing out to me that there was this tree of life, if you will, where there were bacteria or prokaryotes, eukaryotes, which are, animals, plants, etc. And then there was this other group, which it was believed because of the fact that they exist in some rather harsh conditions, the kind of conditions you would have found on Earth in the very early days, that these were the very beginning of life. Well, there was a lot of questions about archaebacteria, and this professor over in the chemistry department was actually growing a specific type of bacteria, which was a methanogen, and it was a strict anaerobe. In other words, any oxygen would kill it. And he was asking lots of interesting questions about this, because back then we didn't have all the genetics that we have today to try to figure out, where on the tree of life archaebacteria fit. But we could ask questions like, how do they get rid of old proteins? So the way eukaryotes get rid of old proteins is they label it with another protein called ubiquitin. But prokaryotes do not do this. So the question then that I got from that chemistry teacher who was really interested in this area was, what role or how do archaebacteria eliminate old proteins inside the cell? And so that seemed really interesting to me. And so that's where I started as far as developing a project. I already known about these things called antibodies. Some of you probably have heard about antibodies, but really quickly, this is the basic structure of an antibody. It's a protein, and at one part of this protein, there is a region which is always different, okay? Because it's all conformation too, right? So these guys have structures and they fit into things, okay? And this structure here will fit into an antigen. Now, we could go into why and how this all works. I'm going to go a little bit, but I just want you to understand that these antibodies are like a lock and key. They stick to things, much like Velcro, for example. Okay, and so these antibodies are actually created by a B cell. Okay, so here's the antibodies being secreted outside. So I'm going to jump back now to make you understand how B cells work. So B cells have this thing called a receptor on the surface. Now, a receptor is simply something we're going to talk a little bit about, by the way, quite a bit actually, because it's the way cells communicate from the outside to the inside of the cell. Okay, in the very end of this receptor just so happens to have the same shape as the antibody. So one B cell creates one antibody. So if, for example, you get an immunization for tetanus, right? Some of your B cells will have this portion of their receptor bind to tetanus. If that happens, then these B cells begin to multiply. And when they do, some of them go into memory, so they can keep those around for a lot longer. But they also produce antibodies to help neutralize that bacteria known as tetanus toxoid. Okay? I'm assuming there's no questions so far, so stop me if anything comes up. Okay, so my project was, I took a mouse. I immunized that mouse with some of the ubiquitin. Back then, you could actually buy ubiquitin, so that was pretty simple. You immunize the mouse, and the mouse saw ubiquitin as being foreign, and some of their spleen cells began, the B cells in the spleen began to activate. And then you can do this thing called a fusion. Using a chemical called PEG, you can actually take myeloma cells, which are B cells, but they're cancerous B cells, okay? These cancerous B cells, you can fuse the membranes with these spleen cells. Now, that's kind of odd because all of a sudden now the cell has two nuclei, those nuclei actually combine, and there's a lot of sorting out of chromosomes that go on. It's a very unnatural thing. But eventually what happens is the cells figure it out and they become a hybridoma, okay? So that's a hybrid of both spleen cells and myeloma cells. What I should have also pointed out was that these myeloma cells, these B cells that are cancerous, they don't secrete antibodies. These spleen cells, however, these B cells coming from the spleen, they do secrete antibodies. And so these hybridomas should have the ability to last a very long time because these spleen cells, if I was just to put them in culture, they would die in about 7 days. But these myeloma cells will live forever. You can keep growing them over and over and over again. Just keep feeding them with media, and they'll grow forever. So now you have the combination of features where you have a cell which will grow forever, and it will secrete the antibodies that you're interested in. Now, you might think about, well, how do you know you're getting spleen cell with a myeloma cell? Well, we use this media with a special media called HAP media. HAP media will kill myeloma cells. Okay, myeloma cells cannot grow in the presence of HAP, but spleen cells can. So the only cells that can survive HAP media are myeloma cells that have been fused with spleen cells, having the combination of characteristics. So these hybridomas then are the ones which have antibodies that you can actually utilize. So you can grow them. Once you can clone them and get individual cells sitting down, and then only you can have one huge culture of that started with a single cell that will produce a single antibody. And that's why it's called monoclonal antibodies. And so the beauty of that is now you have a key which will fit just one lock. And so the goal in my project was, once I've done this, was to find that cell or a couple of cells which are making the antibody for which I was interested in. And so then the goal would be then to do this test known as an ELISA. Okay, so you could also do a Western, but I'm not going to get into that. But an ELISA is also stands for enzyme linked immunosorbent assay. And so the antigen in this case is ubiquitin. I would put this onto a plate, and then I take my antibody, which I've labeled, by the way, with some enzyme, and the result is, if it sticks, and then I try to wash it away, it stays there, and I add a substrate, then the result is you're going to get a color change, right? And that's kind of what you're seeing here in this ELISA plate, right? This is a 96-well plate, very, very common in biotechnology industry, okay? And you can see sort of a change in color that occurs when this antibody with this enzyme is still present. Very similar to, by the way, a pregnancy test or many other tests that we do. There are some other types of ELISAs, an indirect ELISA, where here you're detecting the antibody. So if I was looking for the presence of an antibody, I could detect that antibody sticking to a specific antigen. Or I could have two antibodies detecting an antigen which don't compete with each other, and then and that's called a capture. And then finally a competitive assay, which is a little bit more complicated. I'm not going to get into that, but there are different ELISAs. And so the bottom line in this experiment is that we were unsuccessful because, as it turns out, we couldn't get, there was, in order to grow these methanogen archaebacteria, they grow in the presence of media with yeast extract. And yeast extract is eukaryotic. And there was ubiquitin already in that. And so no matter how much we tried, we could never split apart the yeast ubiquitin from the archaebacteria ubiquitin, if it was present or not. So failure in a sense, I guess. All right, so are there any questions about any of that?
There aren't any in the chat room yet, but please, everyone, feel free to add your questions. Otherwise, we'll keep going and come back to this when you're ready.
All right, so I wanted to kind of go through my science career. It's an interesting career. It's not like most people. So after I went to college and I graduated from UMass, I got my first job at Massachusetts General at this, and Massachusetts General had this brand new building called MGH East. It sits in Charlestown, looking over the Boston Harbor, right in front of the Constitution, and it's a wonderful place to work. As it turns out, I was working in a grant that was written by the director of transplant surgery, the director of children's services, and a doctor on staff in the infectious disease unit. And they were studying this virus known as cytomegalovirus. Cytomegalovirus is a virus which in humans about half of us are infected with. It doesn't cause any problems whatsoever. It is a problem, however, when it comes to transplants. So somebody who is cytomegalovirus negative receives a transplant from somebody who's cytomegalovirus positive, and when you get that transplant, you're going to be immunocompromised, then that's a problem. And so we had a model, whereas amongst other things, we were looking to be able to neutralize cytomegalovirus. We used antibodies, so I did the whole monoclonal thing, as I just showed you, to try to neutralize this virus. But we also were testing some of the early antivirals of the time, like glanciclovir, to be able to see if it would kill the virus. So let me just explain. So a model is something where it's not when you're experimenting on humans, you use something that's like a human and you see if it works there. And our model was pretty interesting. So what we were doing was I would immunize, well, first I have to make cytomegalovirus. So I use a murine version of cytomegalovirus, very similar to the human. And I would infect mice with this virus. I would only give enough so that it wouldn't kill the mouse, but just enough so that they would be what we call latent. In other words, they would have the virus, but it wouldn't cause any harm. And then we would take out the hearts of those infected mice and put them into the peritoneal cavity of mice that had not been infected. And basically, we put this into the peritoneal cavity is the belly of the mouse. And essentially what that would do is that would give the mouse two hearts. And then you could study whether or not that heart would get rejected in the mouse. And it was pretty simple because you could actually see the beating, the heart's beating. And so you could tell whether or not there was some sort of rejection going on by the fact that the heart would stop functioning. So that was an example of a model that I worked on. Following that, I decided to go into industry. So that in Mass General would be considered academia, okay? And then I decided to go into industry. This is a little bit different. Here in industry, we're looking to actually generate products to make money. In academia, you're generally trying to find out information which can be helpful, but not necessarily turn into a product. And so I worked on many different things. One of the most interesting things was that was the period of time when HIV was just coming out, and HIV was we had a way of being able to take T cells and being able to isolate specific T cells, so from a patient, and then we could actually stimulate those T cells to give more. So one of the problems with an HIV infection is that those patients suffer from the lack of CD4 cells. So the idea was that if we could raise these CD4 cells numbers up, they give them back to patients, and make sure we actually cleaned it up. So there were some drugs back then that we could actually incorporate into the culture. And so they would be HIV negative CD4 cells and put them back into patients. So that was probably one of the most interesting things I worked on while at Repligen. Following Repligen days, I stayed in 1 Kendall Square, which is this very large building down in Cambridge, and I went to a company called Variogenics. And here we were looking at, it's a little bit different. I did a lot more monoclonal stuff as I'd been doing. But here we are also looking at the loss of heterozygosity. So what that means is some cancers are spurred on by the fact that they lose part of their genetic material. And we were looking for targets based upon the new only having one of those genes rather than having two sets of genes for a particular disease. Following that, ended up at this company for many years. I was the founding employee at a company called Zykos. Zykos was very, very interested in being able to immunize patients with DNA. See, immunizations today are basically taking proteins from whatever it is that you want to immunize somebody. Like, for example, if you want to immunize people against COVID-19, you take a piece of that virus and then you give it to patients, and then they develop antibodies in an immune system which can recognize that piece of protein and then attack it, so that next time you should get infected by that, then you would have antibodies in an immune system which can respond to that. So, but this is a little bit different. What this company set out to do was to actually take regions of HPV, human papillomavirus, which causes cervical cancer in women. There are many different strains of HPV, and only a handful of those will actually cause cancer. So the idea was, first and foremost, was to identify what regions of the virus and these different strains you would actually have an immune response to. And then once you recognize those, you take the pieces of DNA that code for those regions, and then we immunize patients with that. And of course, we had an animal model which was intended to do just that. We could show that it worked, and it was very, very effective. Okay, but then there came a day when, because I adopted a child with special needs, that I needed to do something other than research. I lived in New Hampshire, and so I've always thought about becoming a teacher, and this seemed like a great opportunity. And what I found when I went to Milford High School was that they had a real biotech. It was the first school to actually be set up to be able to teach biotechnology. While there, I started the first science fair at the high school, which would later, the following year, lead to a statewide science fair. So the New Hampshire C, me and the original teacher in biotechnology, who had moved over to the Seacoast School of Technology, we got together after the first year and realizing how successful it was at Milford High School, and decided to make this a statewide event. Realizing that in teaching biotechnology, that you wanted students who you need to sort of complete the cycle, right? So you had students who were doing science at the high school level, and some amazing science, I have to say. And we wanted them to be able to present that to outside people. And so we generated this whole statewide competition, which still exists today. Yeah, it was an awesome period of my life. And back then I used to teach microbiology, biotechnology, as well as anatomy, physiology 1 and 2. So I was getting all the students who were interested in careers in science. And it was very exciting because students at this level are, you can really turn on students to these types of careers, and it's really been satisfying seeing all of them and how successful many of them have been in their careers. But as all good things have to end, I always missed working in the lab, and so that's why I'm currently at MustangBio. I've been here for about a year and a half now, and one of the things that I've been asked to do was to develop, we have a program called XKID, and I wanted to talk a little bit about that. And I wanted to get into a scientific presentation. So any questions so far?
In fact, we do have a couple questions.
We do.
Yeah. So question from Pablo, which is, what constitutes the color change in the ELISA? Is there a specific indicator used? And I think that was back on slide 15, maybe.
Yes. So, okay. So let's see if I can find that really quick. So in the ELISA, okay, there is a specific enzyme. Right. So here, so the question was whether or not you could change so there are different colors that, it depends on the enzyme, right? So the enzyme that you put here, okay, it's clear, right? The liquid will always be clear, but then when you add the substrate, the enzyme changes the substrate, which is also clear when you first put it in, to change its chemical property so that it actually has color. And that's it. So there are different substrates that can produce different colors. But here's the interesting thing, is that the more there's more of this around, the darker the color will be. Okay, so you can actually use that as a way of being able to measure how much your antigens you're actually measuring with this one antibody. So the more antibodies that stick there, the more enzymes you have, and the more of the substrate that will get converted and change color. I hope that answers the question.
I do not see any follow-up, so we will assume it does.
Okay.
I'm going to also unmute Balaji, who had a question as well.
Yeah, so I think my question was on the previous slide. Yeah, so you stated that myeloma cells had the quote unquote like trait to live forever.
Correct.
And that trait is inherited in the hybridomas. But if the hat medium kills the myeloma cells, does it not take away the properties of the myeloma cells as well?
No. So a little bit more detail. What HAT is, it's a way of being able to generate energy, right? And it's a sort of a side pathway that only spleen cells can do. Okay, so you make these new hybridomas have to utilize that pathway. And so the only way they can do it is they receive genes from spleen cells. You see, because when you do the fusion, you can get myeloma cells fusing together. You can get spleen cells and spleen cells fusing. This is not something where you're taking one cell, a myeloma, and a spleen cell, and putting them together and making a hybridoma. It's not like that. You don't have any control of that. All these fusions are occurring at the same time. And so you have to have a means by which to separate out where you got a myeloma cell fusing to one spleen cell to give you this hybridoma. And all the rest of them will be killed because they can't utilize the HAT media. And so what you're left with is a bunch of cells which are antibody producing, and they will live forever.
Okay.
The goal then is to basically spread these out on one of those 96 well plates, trying to put one cell per well, and then you let them grow. And so one cell will populate into a large colony, which will then, and if you find that that colony is producing an antibody that you're interested in, just going back, that's where you actually, whoops. And so that's where I would use this assay, right? So I put my antigen down. In this case, it was the ubiquitin, and I put in some of these different, each well. I take the material out and I put them in, and I want to see if there's an antibody there that sticks to that. And then I wash everything away. If that stays there, Then I add a secondary. So this secondary antibody is one that's specifically made in rat. So rabbit was immunized with mouse. Okay, so this rabbit antibody will stick to mouse antibodies. Okay, and that rabbit antibody you can buy that already has an enzyme on it. So this is an indirect way of being, it's an indirect ELISA, and that's the way you could tell which one of these wells actually has cells which are producing this antibody, which sticks to ubiquitin. See, there are many of them will not. So when you look at all of these, not all of these cells will stick to ubiquitin. The mouse could have been, you know, they're going to have other B cells in there. Not all of them are going to be attacking the one that you're interested in. So you have to sort of sort through that. And that's called cloning.
Okay, thank you.
Yep.
We have a question from Nikki. I'm going to try to unmute here. Go right ahead.
Oh, I'm not sure. Oh, does my mic work?
Yep, we can hear you.
Okay, great. I was just wondering if there are any negative consequences that like arise from using the cancerous B cells, the myeloma cells.
Negative.
Like, because I would assume, you know, since they are cancerous,
Yep.
just wondering if any problems would.
No. As a matter of fact, I actually had students in high school actually using these because they can't infect you, right? It's because they're mouse cells. Your body, if you were to drink a bunch of myeloma cells, you're going to reject those and kill them just like you would if you ate a piece of mouse meat, if you will. And so they're only a problem to mice. Growing human cancer cells is different, right? So if I was growing human cancer cells, that's where you have to be careful for sure.
Yeah, okay.
Good question.
Thank you.
Any others?
No others right now. I will keep you posted.
Okay. So now I kind of wanted to get a little bit more technical. And this has to do with what I'm doing at Mustang. So at Mustang, we do two things. We have a CAR T program, all right, and what CAR T is we take out cells out of patients, particularly blood cells, and we isolate specific T cells. T cells are sort of the part of your immune system which is responsible for hand-to-hand combat. Like B cells, I've always described as archers. B cells, which I showed earlier, they make antibodies. So they're basically shooting out antibodies into your entire body. They're like archers in this war against things that are foreign, right? Because they can sit in a spleen and sort of shoot out their antibodies and they'll hit their targets. Different than that are T cells. T cells are like the hand-to-hand combat. They have to actually make contact with another cell, which is foreign, like a cancer cell, to actually kill it. Now, what happens when somebody has cancer is that over time, we probably all get cancer at some point. And the good thing is that lots of times our immune system can take care of that. The downside is that sometimes it can't. And so what CAR T, this is just developing over the last, say, 7 years, has been really big at being able to, it's called personalized medicine. You take out cells out of a person, a patient, and you isolate specific T cells that you're interested in. You put a gene using usually virus, a lentivirus, into those cells to reprogram that cell to specifically target the cancer cell for which that person's been afflicted. You put them back into that patient and those T cells will go and kill those cancer cells. Okay, so that's called CAR T. I haven't worked that much with CAR Ts at this point. I am in the coming year, but this past year, I've really been working on XSkid, another type of gene therapy. Now, somebody who suffers from XSkid, you might remember, well, some of you might not be old enough to remember the John Travolta version of the bubble boy disease, but essentially these kids are born without a functioning immune system. and it has to do with a receptor, and I'll talk about that in a moment. But it primarily it's X-linked. So from the mom, these boys are receiving an X chromosome, which has a defect in this particular gene, which I'm going to talk about right here. So it primarily affects just boys, although there probably are a few girls out there. It's a rather rare disease. So we're talking about 20, maybe 30 patients a year in the United States. But it really, for us, is a proof of concept, if you will, a way of being able to show that we can actually do this, where the goal here is to take lentivirus, right? So we take out the bone marrow out of these very, very young babies, okay? And we take out bone marrow because we want to get these developing cells. So, all right, so let's step back for a second. In your bone marrow is where all of your blood cells originate from. These are called hemopoietic stem cells. These hemopoietic stem cells can become any blood cell in your body. It can become a white blood cell, a red blood cell. And then amongst the white blood cells, it could be a CD4, a CD8, you know, et cetera, right? So that's where all of these cells come from. So we want to get these cells that haven't differentiated. They have the potential of becoming any type of blood cell. And so specifically, these cells are called CD34 cells, but I'm not going to get into that right now. So what I did want to talk about was, let's just talk about the concept of what a receptor is. Okay, I kind of hinted at what that was before. Okay, essentially a receptor is something that goes through a cell membrane. So here's your typical, you know, phosphohead lipid bilayer. Okay, right. This happens to be the outside of the cell. This is the inside of the cell. Right. Receptors go all the way through the membrane somehow, and they're anchored in the membrane. Something happens on the outside of a receptor, which then signals something on the inside, right? And of course, that doesn't make that, what that means is that somehow it goes to the DNA and turns genes on or turns genes off, okay? So we're talking about a pretty complicated system here of biochemistry. But just understand that that's what we're going to talk about here. We're going to talk about receptors, which bind to these things called ligands. All right, not all ligands fit into every receptor. So you have receptors that recognize cytokines. So cytokines are messengers from other cells. It's the way cells can communicate. You have cytokines like IL-2, IL-4, IL-8. All of these interleukins, for example, are cytokines which are secreted from another cell trying to communicate. So this is like the wireless system in your body, okay? So you have a wired system in your body called nerves, right? But you have this wireless system in your body, in your immune system, to be able to communicate. So if some cell has found some foreign invader, it wants to be able to communicate, you know, that I've engulfed, for example, a bacteria, and it's going to release some cytokines to tell other cells to get going that we need to go and attack this, okay? So, all right, so hopefully we got that concept where a receptor is. And so a little bit more detail here, okay? So here is an example of a receptor that we are working on, okay? And so specifically, one of these receptors, so these cytokine receptors that we work with, one of these is doesn't work, okay? And essentially the cytokine receptor sits there and it binds a cytokine, that signal from that ligand from another cell, right? As a result of that, it brings together these JAK proteins close together and it phosphorylates them, which then triggers a phosphorylating event on the receptor, which then triggers this STAT 1 and 2 to actually get, and STAT 3 and STAT 5, all these different stats to also get phosphorylated. Don't want to get you bogged down in all the details. This is biochemistry, right? So this is the stuff where people have studied, you know, many, many years to try to figure out how this system works. Why is that important? Because understanding how it works, you can understand when it goes wrong. But here's the important thing, what I want to talk about. let me finish this off by saying that this STAT molecule, once it's been phosphorylated, goes into the nucleus, sits down on a gene, and that's what turns the gene on. In other words, whatever cellular response that cytokine, this cytokine here, that started it all, this is what's going to turn that response on. And whether that means producing more cytokines, whether it means to get more activated and start proliferating, there are many different types of signals that can turn on different genes. Okay, so when one of these is defective, okay, this doesn't work. And so I, in this past year, have targeted specifically this right here. I'm very fortunate that somebody actually made an antibody, okay, which recognizes this phosphorylated form. So if this phosphorylation has not occurred, the antibody would not recognize this. So I have an antibody which specifically recognizes just that, okay? And so what I want to show you here is that here are 6 different cytokine receptors, right? And here is IL-2, IL-4, IL-7, IL-9, another form of IL-2, and IL-21. What you're going to see here amongst all of these is that what makes them different is the alpha chain, but what's common amongst all of them is this chain right here. All right, it's the gamma C. This has a gamma C. This has a gamma C. So half of the receptor is this gamma C, and it's this gamma C portion of the receptor which is actually broken. Okay, and so that's what these XSCID patients suffer from. And so without having receptors that work, you don't get any phosphorylation occurring, and essentially these immune systems just sit there and they can't. It's like having a phone without a receiver. You can't hear anything. So how do you give a response? Hope that was a good analogy.
So, Rich, I'm sorry to interrupt.
Yeah.
We are now almost 5 minutes past our time, and there's a lot of rich content here to cover still.
Yeah.
I'm wondering if perhaps the students on the call perhaps would be willing to find another time for us to continue the conversation, because you've all stuck with us, which shows your interest, which I'm really excited about. And I know, Rich, you've put a lot of time into preparing for this.
Yeah.
So what I'm going to suggest is if we could, if you'd like to wrap it up,
Okay.
and perhaps everybody on the call, because we didn't have a formal registration for this, anybody could click through. If you could all email me at info@scifair.com, that way we can find another time to complete this and allow you some more Q&A time. Would that work?
Yeah, that's fine with me.
I want to just be sensitive to everybody's schedules, and I know you've put a lot of time and effort into this, and it's really clearly interesting to our students. So I will just turn it over to you for if there's a natural wrap-up spot before we complete our session.
Yeah, I think if I was to wrap this up, I mean, really what I was going to talk about was, what cells are affected, how I go about being able to see these cells in a particular type of manner. It's called a flow cytometer, how we use antibodies. This is a flow cytometer machine that can actually just, read what I'm doing. I had a little video, which we can show in the future. I can show some data. And if I was going to sum it up right here, I just want to show that, I've got this thing working. I can actually see, in the case of IL-2, I can actually show it. I can see that phosphorylation. Here's some more real data where IL-4 works even better. And then I was also going to talk about positive controls. IL-3 does not have a gamma. So these two both share gammas. This one is not a gamma, but it does signal through phosphostat 5. And so it works really well. And then I also wanted to show some cells where ED7R cells, these cells have been stripped of the gamma C. And so it's a really good model for being able to look at whether or not this would work. And so be able to take our virus, put it into, and you can see that in fact, it does retain some function. And so I guess the bottom line was, this therapy does work. We've had it into patients. We actually licensed this technology. We licensed this from St. Jude down in Tennessee, but they're an academic. And so we need to be able to blow this up into more patients, be able to, and to be able to treat more patients. So our first patient is expected fairly soon for XKID, that we will take over the treatment from St. Jude. So it's really exciting times in gene therapy. Yeah.
Well, great. Thank you so much. I'm really sorry we had to kind of wrap it up so quickly. But I'm thrilled that so many people are interested in this. And I, again, want to just repeat to those who have called in that if you email info@scifair.com and indicate your interest in continuing the conversation and learning more, we'd be happy to coordinate another session to whether it's just a few of you or the whole group to talk in more detail. So
science projects.
Yeah, for sure. So thank you, Rich. Really appreciate your participation today. Thanks to all of you who called in. And our speaker series continues next Tuesday through Thursday at 3:30 every day. So you can find the list of future speakers on our website. And thanks all for participating. Appreciate it.
Thank you.
Have a good afternoon.